Ooh,
Unmute.
okay. Can you guys hear us now? Let's see. Sorry, just one sec. Chat.
Yeah.
Yes, okay, we're good. Cool. Okay, yeah, so this is our big ugly picture that we're gonna kind of keep coming back to. This, I think, was in a couple of your guys' lectures with Dr. Sukup. He really likes this picture. He likes to ask things about where different processes take place. So if you're in the mood to stare at this for a while, I would say do it. But we're gonna keep talking about it too. So you can go next. So we're gonna start off with just like glycolysis, okay? And Cassie's gonna take it away.
Okay, so this seems like a low yield slide, but it might be good to know. My memory is foggy, but I do remember stuff like this. So just understanding kind of why it's important to have transporters that are active all the time versus ones that are only active at high concentrations. And I know in your guys' lecture, there was like a graph showing it, but it's pretty much just like at normal basal level, you have GLUT1 and GLUT3 transporters working all the time. And then whenever you have higher glucose states, that's whenever GLUT2 will get activated. And so that allows for more transport in like a concentration dependent manner. So I would make sure that you understand kind of how KM relates to that. So a higher KM means that it's not going to be active unless there's a higher glucose concentration. Does that make sense to everybody? Pretty self-explanatory. All right, so glycolysis.
There's a lot of memes, so just get ready.
And if you don't understand them now, look back at them on Thursday night and you'll understand them.
It'll be worth it.
So this is the big ugly pathway of glycolysis. And we are going to talk pretty much about the three main steps that are important in all of this. So if you want to memorize everything on this, you can. I recommend focusing your time elsewhere. But first things first, we're going to go through some questions. So if you guys are brave enough to answer, that would be really exciting because I'd rather not sit here in silence. So does anyone know where glycolysis happens in the cell? Yeah, go for it. Cytoplasm, that's important. Know where every different metabolic pathway takes place, whether it's in the cytoplasm or in the mitochondrial membrane or in the mitochondrial matrix. Make sure you guys are paying attention to that because it can trip you up on questions. So yes, cytoplasm. Does anyone know what the committed step is, not for glycolysis, but just in general for glucose being trapped into the cell? What? G6P, yeah, so hexokinase. So hexokinase is what converts glucose to glucose 6-phosphate. And then once it's glucose 6-phosphate, it cannot leave the cell, which means that it's destined to do some sort of metabolism somewhere in the cell. So apart from the committed step, what is the rate limiting step of glycolysis? Do we know that one? PFK1, the best enzyme. That's not even a question. But yeah, I just told you the answer to that one. I'm really sorry. Okay, what's the net ATP? Does anybody know that? 2. So you start with 2, you use 2, you gain 4, net 2. So pretty cool. So do you think you want to add to that?
I don't think so. No, just, yeah, plus 2 ATP. And just remember that lactic acid is produced to regenerate your NAD plus, okay? Because I think it's glucose 6-phosphate dehydrogenase that requires NAD plus to function.
Yes, so. And also glyceraldehyde 3-phosphate in the glycolysis pathway.
Maybe that's what I meant. Yeah.
Yeah. So understanding how NAD is regenerated via the lactic acid pathway is good to know as well. So I would highly recommend understanding that. Okay, so the 3 key enzymes, we said 2 of them. What's the third one? Does anyone know? Pyruvate kinase. Good job. You guys are so ready for this already. And then we're going to go through key stimulatory and inhibitory molecules. This is also very high yield. I would understand how different substrates will either activate or inhibit enzymes and pathways in general. So first, let's go with hexokinase. So the number next to it, like the 1, 3, 2, that's how many things there are. So does anyone know what will inhibit hexokinase? G6P, yeah. So which makes sense because, you know, product will inhibit the enzyme because you don't want to have too much of a buildup of the product. Does anyone know what will inhibit PFK1? Does that inhibit it or activate it? Activate it. Good job. I'll ask you again in a second. Does anybody know? ATP, yes. I heard citrate, and then there's one more that's kind of weird. Low pH. You guys are good, good.
You guys are crushing it.
Okay, stimulate. We got one. What is it? PFK2. And then anybody know what the other one is? AMP. Why isn't it ADP? ADP is just bad at telling if there's energy in the cell or not. So also remember that AMP is what will tell you if you have low energy in the cell, not ADP. That's important to know as well. Yes. Yes. But for glycolysis, it's AMP. And then lastly, pyruvate kinase. What inhibits pyruvate kinase? What was that? There's one kind of weird one that's random. Alanine, yeah. And then the other one? ATP. And then what will stimulate pyruvate kinase? Yes. So it's kind of like a feed-forward mechanism. The more fructose 1,6 bisphosphate you have, the more activity of pyruvate kinase you'll have because you don't want to have a buildup of product again. So now we're going to talk about this lovely step, which is the best step of glycolysis. So it's important for you guys to understand the regulatory, sorry.
No, you're good. I'm just...
Understand like this regulation of PFK1. So this is kind of a good slide, but the next slide's a little better. But pretty much, so you have fructose 6-phosphate. That is a product of glycolysis, and it can either go forward to fructose 1,6 bisphosphate via PFK1, or you can have an enzyme called PFK2 that will convert it to fructose 2,6 bisphosphate, which is what we said will activate more PFK1. So it's kind of like a side pathway where if you have too much fructose 6-phosphate, both enzymes will be working together. So you'll have PFK working really hard to get as much fructose 1,6 bisphosphate as it can, but at the same time, there's so much fructose 6-phosphate that there's going to be a side pathway that's going to enhance PFK1 more. Does that make sense? Does anyone have any questions about that? Okay, and then the other thing to know is that PFK2 is closely linked to FBPase2, so it's a complex, and so it's regulated via phosphorylation and dephosphorylation, which is what we're going to talk about next. So this is also a very important slide to know. So once you guys like understand kind of the regulation of this, just kind of run through it and be like, okay, yeah, that makes sense. Because I would look at these kinds of like figures when I was studying last year, and I was like, this does not make sense to me. But once you kind of just like take it step by step and then put it into big picture, it'll be so much easier. So before we go through the really nitty gritty things, I think it's important to know glucagon and insulin. So do we know which one phosphorylates and which one dephosphorylates? Which one phosphorylates? Glucagon. Therefore, insulin dephosphorylates. Just by knowing those two things, that will help you so much. It helps you to have to memorize so much less. And if you know what glucagon and insulin does, so like glucagon is whenever there's low glucose, insulin is when there's high glucose. So the way that it works is glucagon is a phosphorylator. So it goes through the whole GS pathway of cyclic AMP and PKA. And then that phosphorylates the PFK FBPase2. And then that means that we don't want to go through glycolysis. We want to conserve as much glucose as we can, right? So phosphorylation of the PFK2 FBPase2 will stop glycolysis, or at least tone it down. It won't fully stop it. And then insulin will do the complete opposite. So say that we have lots of glucose, then dephosphorylating this complex is going to activate the PFK2 part, which is going to up glycolysis, get rid of more glucose, break it down into different kinds of things. Does that make sense? Anyone have any questions about that?
Yeah. Insulin is secreted when you have high blood glucose, right? So if you have a lot of glucose, it makes sense that you're going to want to do some glycolysis to lower that blood glucose down. Okay.
Exactly. And so the glucagon and insulin being phosphorylators or dephosphorylators is going to come back up again in different pathways. So just by knowing that, you can kind of reason through what's going to happen or when each enzyme is going to be active. If it's phosphorylated, is it active, or is it dephosphorylated when it's active, which we'll kind of point out as we go. But yes, this is just a good slide.
Yeah, just for your reference, like we said, you don't have to know every single step, I don't think. Just focus on the 3 that we talked about.
I agree. And also I would know the 2 steps that generate ATP. One of them's the pyruvate kinase, but the other one is, what is it? The 1,3-bisphos, yeah, the phosphoglycerate kinase. I would just know that. I don't know if they asked about it, but just knowing where ATP is generated in general is pretty important. Okay, take it away, Dan.
Okay, all right. So gluconeogenesis, right? So that's my, do you guys like Lord of the Rings? I do. So I put this on there. So anyway, so we're going to kind of go through the same thing here with gluconeogenesis. Okay, so we're going to start off. Where does gluconeogenesis occur? Which organ? Do we know?
You can shout it out.
Just shout it out, yeah. Liver, very good, liver, yep. Where in the cell does it occur? Cytoplasm, good, very good. And it also does a little bit in the mitochondrial matrix. Good, the first couple steps. Very good, okay. So why does it do this? Why is it partly in the matrix and partly in the cytoplasm? Does anyone have any ideas why? It's not super intuitive, yeah. Very good. Yeah, it has to do with the electrons. So you got to get your electrons out of the mitochondrial matrix and then use them for that glyceraldehyde 3-phosphate dehydrogenase step, which, if we recall, required NAD+ going through glycolysis, but now we're going the opposite way. So we need NADH. Okay, so we got to get that NADH out from the matrix into the cytoplasm. Okay, so that's our step, right? This is the key here. So you can see that it requires NADH. Okay, so there's a couple different ways you can get this NADH out of the matrix into the cytoplasm. The big one is probably this malate shuttle here. So you convert oxaloacetate to malate, which is then transported out, converted back to oxaloacetate, yields NADH. Okay, the other way, which I think is more interesting, is this here. So converting lactate to pyruvate also yields NADH. Okay, so there's this thing called the Cori cycle, where this is what happens in exercise when your muscles run out of glucose. Okay, so you have your muscle breaking down glucose, yielding lactate, right? Because we're running out of oxygen. We need to do anaerobic respiration. So we're getting a ton of lactate in your muscles. So what you can do is you can send that lactate back up to the liver, and then your liver can do gluconeogenesis starting from lactate. And because of that, we get to skip this whole malate nonsense, and we just get to go straight through, okay? Because we get our NADH right there. So then we get glucose back, gluconeogenesis. We ship that glucose back to the muscle, okay? And then we just do the same thing. So that's the Cori cycle. That one kind of makes sense. It's pretty intuitive, which is I kind of like. So not everything in metabolism is. So okay. So next question. So we kind of talked about this already. Do you remember the 3 enzymes in glycolysis that are irreversible? Yeah. Why are they irreversible? Why is this the case? Do we know? Very good. Negative delta G. Yep. A lot of free energy is produced by these reactions. So you can't use the same enzymes to go in reverse. It's too energetically unfavorable. You have to use different enzymes. Okay. So yeah, those are our 3 enzymes. Okay. The last step here. So where's glucose 6-phosphatase located? Does anybody know? It's in a weird spot. ER. Very good. Yeah. Why is it there? Good. Yeah. So you need to separate glucose 6-phosphatase from hexokinase because they're doing the opposite thing. So if you had them right next to each other, they would just undo each other's reactions constantly. But by isolating glucose 6-phosphatase into the ER lumen, you get to separate those 2 reactions. So
I think it's important to know that. So just put a little star next to that.
Yeah. Okay. So what is the net energy use or production in gluconeogenesis? Very good. Yep, exactly. Okay, so now we're going to talk a little bit about regulation. Okay, so it's kind of the same thing as glycolysis. It's the 3 same enzymes, 3 same steps, just in reverse that are regulated. Okay, so we're going to do the same thing here. So what inhibits pyruvate carboxylase? ADP, very good. Okay, what about what stimulates pyruvate carboxylase? Think about where it is in the pathway. Acetyl-CoA, very good. That's a reactant, so it should make sense, or it's close to a reactant. And then PepC-K, it only has an inhibitor. What inhibits that? ADP, very good. Okay, same as pyruvate carboxylase. Okay, oops, I gave it away. What inhibits fructose 1,6 bisphosphatase? So, yep, AMP, very good. What's the other one? 2,6 bisphosphate, very good. So that's the opposite, right, of PFK1. So that should make sense because we don't want gluconeogenesis and glycolysis occurring at the same time. So that should make sense. And then lastly, what stimulates fructose 1,6 bisphosphatase? Citrate, very good. Yeah, citrate is part of the TCA cycle. So if you have a lot of citrate, you have a lot of carbon skeleton to work with to make glucose. Okay, so very good. You guys are crushing it. Is this yours?
Yeah, this is mine.
Okay, yeah.
Okay, so we're going to talk about glycogenesis, whatnot. So first we're going to talk about, this is just, we already labeled them for you, but knowing how to tell the difference between a reducing and non-reducing sugar is a good skill to have. I would make sure you guys kind of understand what makes a sugar reducing versus non-reducing. Obviously, yeah, I already labeled them for you. Okay, so what are the two types of glycosidic linkages in glycogen? 1,4 and 1,6. Which one's linear and which one's branching? Nice. I know those are kind of easy questions, sorry. All right, and then which enzymes are responsible for each of them? Well, let's do the 1,4 linkage first. Which one is that? Glycogen synthase. And then what about 1,6?
Branching enzyme, very good.
The easiest part of science. Okay, so what's the importance of branching? Why do we branch it? Why don't we just make one really long glycogen? Yes, so you have multiple different ends that you can access glycogen. So whenever you're breaking glycogen down into glucose, that means that you are you're depleted of glucose already. Like you need more glucose. So if you were just to break it down one at a time, it'd be very inefficient. So by branching and having a bunch of different non-reducing ends, then that allows you to get more glucose faster. So yes, it takes it from the non-reducing end. Make sure you guys know that. And then which enzymes are responsible for the opposite? Debranching, yeah. And then what's the other one? Yeah, good job. So why do we need to debranch? Why can't glycogen phosphorylase do it all? What's the purpose of the debranching? It's kind of a hard question. I worded that wrong, but pretty much glycogen phosphorylase cannot break up 1,6 linkages. So you have to first transfer it to a 1,4 linkage, and then glycogen phosphorylase can then do its job. So this is what we were talking about earlier with the glucagon and the insulin. So if you know which one phosphorylates and which one dephosphorylates, which one's released with high glucose versus low glucose, then you can pretty much reason through this. So which enzyme is activated by glucagon? Just based on the pictures up there? Correct, phosphorylase, because that means that you're breaking glycogen down to get glucose, and glucagon is low glucose, so that kind of makes sense. Glucose, yeah.
That's my favorite acronym. That's how I remember it. So if it works for you too, that's good.
What about, which one is inhibited by glucagon? Yeah, pretty much just the counterpart. Pretty self-explanatory. And then which one's activated by insulin? Synthase, because insulin means high glucose. So you want to store more glycogen because you don't want to break any glycogen down. And then same thing, you guys know what this is. Do you want to talk about this slide?
Yeah, sure. It was shaking for a second. It was freaking me out. Okay, so this is just a good summary slide of all the regulation of carbohydrate metabolism. So take a look at this. We've talked about most of these here, but just go through this and work through it. And this we'll let you do on your own time, but we got a lot of other stuff to get to, but good slide, okay? Bottom line, so take a look. Okay. This is you again.
Yeah, this is my favorite thing ever because the pentose phosphate pathway looks so complicated, but really you only need to know like 3 things from it. So first, which one is the rate limiting step? Yeah, glucose 6-phosphate dehydrogenase. I would make note of that and know that pretty well. And then what are the products of it? Oh, I didn't ask that question. Sorry, I also accidentally just showed you guys the answer. But yeah, NADP and G6P are the activators of G6PD. Where does it occur? The cytoplasm. Good job. And then what are the products? I asked you this a couple seconds ago. Perfect. Oh, what's ribose 5-phosphate used for? Yes. And then what about NADPH? Perfect. You guys are good at this. You could take the quiz now. And then what's the ultimate function overall? I'll just show you because we just pretty much talked about it. So you pretty much, the whole purpose is to create NADPH that can trigger more different pathways. So the pentose phosphate pathway generates things that are going to go to all different kinds of pathways. So it's kind of just like an intermediate.
The two main ones with NADPH are fatty acid synthesis and cholesterol synthesis. Okay, those are the two where NADPH is used. So we'll go over that too. Okay, this is just a table you can look at. I don't think that's super important. Okay, so this is me, right? Yeah, okay, all right, so pyruvate dehydrogenase. So they love this enzyme, okay? They will ask you a question about it, I guarantee it.
Be more than one.
Yeah, you might get 2, you might even get 3. So here we go, pay attention. Okay, so let's see here. Okay, yeah, so which enzyme is involved in conversion? So yeah, this is what we're talking about, right? Yeah. Why is this even on here? Okay. Okay, where is this occurring? That's important. Very good. Yeah, pyruvate gets transported into the matrix and this occurs. Okay. Good. Okay, so there are 5 cofactors for this enzyme, and I don't know why this is so important, but it seems to be asked a lot. Okay, so we're going to go through the 5. Can anybody list any of them before I go to the next page? Thymine, good.
I heard niacin.
Okay, good. Good, good. You guys are crushing it. Yeah, you guys already know this. So those are the cofactors. There's 5 of them. And I really don't think you need to know where each of them act, but I would know that they're necessary. Okay, a lot of B vitamins. Okay, 1, 2, 3, and 5, and then lipoic acid are all important. Okay, this is important too. Okay, so there's a lot of regulation of this enzyme. Okay, so we're going to go through all the different regulators, and you're going to tell me if they stimulate or inhibit PDH. Okay, acetyl-CoA. Good. ADP. Good. Good. Low energy state, right? ATP. Good. Calcium. This one isn't as intuitive. Good, good, right? Because we release calcium when our muscles contract, so we need to be sending things into the TCA cycle, okay, to get more energy. How about insulin? Activate, good. NADH. And pyruvate. Very good. So that's it, okay? There's seven of them. It's kind of crazy, but if you think through all of them, they should all kind of intuitively make sense. It's just like, you know, there's just 7 of them, so that's a lot. Okay, so this is kind of the regulation just grouped into different categories. So there's 3 main allosteric inhibitors of PDH, which are acetyl-CoA, NADH, ATP. Now, the activators of PDH don't act allosterically. Okay, they act by this enzyme called PDH kinase. So, or they actually, yeah, they stimulate PDH by inhibiting PDH kinase. Okay, so because PDH with a phosphate on it is less active. Okay, so by inhibiting this enzyme, we're actually activating PDH. Okay, and then on the other hand, we have calcium and insulin, which stimulate PDH through activating the phosphatase. Okay, remember insulin takes phosphates off, so we remember that. Calcium also does that in this case. Okay, so removing that phosphate activates the enzyme. Okay. So, okay, is this yours? Still me? Okay, this is the real Krebs cycle. So I've been through it a few times. So anyway, okay. So we're going to try something a little different. So I'm going to point to this thing and you're going to tell me which substrate goes in the box or what goes here. Nice. How about here? Very good. And then here, oxaloacetate, very good. Okay, so what does this yellow star represent? What is produced at these locations? Very good, NADH. How about this purple little thing here? GTP, very good. How about the orange diamond? FADH2, very good. Okay, yeah, this is important to know. Okay, I would say definitely, if you don't know that already, stare at it. So let's label the steps in which each products are produced. Okay, yeah, so we already did that. So what is the purpose of the TCA cycle? Like what electron, right? We're making NADH and FADH too, but mostly NADH, okay? That's the big one, because that one is a little bit more energetic than FADH2 and getting those hydrogens across that inner mitochondrial membrane. Okay, so I just wanted to briefly remind you guys citrate here, how it interacts with carbohydrate metabolism. Okay, so remember that citrate inhibits PFK1 and stimulates FBPase2. Okay, so it inhibits glycolysis and stimulates gluconeogenesis by those interactions. Okay, it's an important one to know. Okay, so just briefly, regulation of the TCA cycle. Most of these are intuitive. Once again, it's just sort of either products stimulate or products inhibit, reactants stimulate. And I would say also the important thing to know here is they love to ask you succinate dehydrogenase, how it's complex 2 in the electron transport chain. That is, I don't know why that's, yeah, they love to ask it. So just know that, okay? That's a little factoid that'll get you a point. Okay. So this is just an interesting side point here. So if you're running low on oxaloacetate, you can actually convert pyruvate directly to oxaloacetate via pyruvate carboxylase. So this is sort of a shunt that avoids acetyl-CoA if you're really low on carbon chains and you need to go through the TCA cycle, okay, and get you NADH. So this little enzyme is maybe important to remember. Pyruvate carboxylase. Okay, fatty acid oxidation. Okay, I love Star Wars, so yeah.
Before we start, does anyone have any questions?
Yeah.
We good?
How are we doing? Hanging in there?
Cool. All
right, how are we doing online? Someone respond so I can. Golden, I love that. Okay, we're chilling. All right, fatty acid oxidation, okay. What the heck? There we go. Why did that do that? Okay, well, that didn't work. So anyway, so where is fatty acid oxidation occurring, right? This occurs in the mitochondrial matrix. Okay, we have beta oxidation in the matrix. So how do we get fatty acids into the matrix though? Okay, we use this thing called the carnitine shuttle. Okay, we use carnitine because fatty acid bound to coenzyme A, that just is it's too bulky. It can't get across the membrane. But if we remove the CoA and bind the fatty acid to carnitine, we can use this translocase here to move that fatty acid into the matrix, and then we can rejoin it with coenzyme A.
I would make sure that you understand CAT1 and CAT2 and the translocase and the purpose of it. They probably stressed it in lecture. They stressed it to us too. And whenever they stress stuff, you know what happens.
Yeah. So so CAT1 is the one that does it in the cytoplasm, and then CAT2 in the matrix. Okay. So this is just a picture. I don't know if you guys have ever been on First Aid. I would highly recommend getting on there, and they have really good little blurbs, articles about these sort of things, and they really help me. So if you're interested, they have good figures too. So this is one from First Aid, just showing the carnitine shuttle. So we have CAT1 out here. And then CAT2 in the matrix, okay? And then we have this transporter. So we already kind of did this. So which one does it in the cytoplasm? Cat 1, good. What about transporting acylcarnitine across the membrane? Good, translocaser, it's labeled transporter here. Okay, and then what about regenerating fatty acyl-CoA in the matrix? Cat 2, very good. Okay. So what's the major regulation site now of beta oxidation? I hear Cat 1. It's Cat 1. Okay, Cat 1 is your regulation site. So what inhibits Cat 1? There's really only one thing. You can see it from the picture. Good, yeah. Malonyl-CoA. Okay, why? Why is malonyl-CoA inhibiting Cat 1? Very good. Yeah, malonyl-CoA is it's after the committed step of fatty acid synthesis. So if you're doing fatty acid synthesis, you have a lot of malonyl-CoA, you don't want to be doing fatty acid oxidation at the same time. Okay, so it's sort of a, they inhibit each other, the pathways. Okay, explains there. Okay, they like you to know the order of this, the adding of the breaking down, excuse me, of the 2 carbon chains. So there's 4 reactions that take place to break off 2 carbon units in acetyl-CoA. Okay, so first you do a dehydrogenation, okay, hydration, dehydrogenation, and then cleavage, dialysis. Okay.
And it's important to know the order of them too.
The order is important. I wouldn't say you need to memorize the names of these, but I would say the order is more what's important here. Okay. So we're cleaving 2 carbon units off of our fatty acid chains in acetyl-CoA. Okay. Any questions about fatty acid oxidation? Yeah. Do you know?
Dehydrogenation. Sounds like it.
Well, no, it's hydration. So I think that more just is adding a water. So I think it might be reduction. Well, no, it wouldn't be reduction because you're oxidizing. Okay.
Okay.
So that wouldn't. Okay, yeah. So yeah, dehydrogenation, oxidation. It wouldn't make sense to reduce it after. So I think this is different from reduction. But yeah, if that's what they were saying in lecture, that makes sense because you're oxidizing, right? Overall. So yeah. It's cleavage of the. Yeah, it's cleavage using like a thiol group. So a thiol group is a sulfur hydrogen group, and you're using that to cleave 2 carbon unit off. So it's just another word for cleavage.
Yeah, so you have the CoA SH, and then you have the thiolysis, and then that generates the acetyl-CoA right here, right there.
Cool. Good questions. Anyone else? Cool. Okay, so this is sort of yeah.
Oh, I think that was just them saying golden. Chilling.
Chilling.
We love to hear it.
We love that. Okay, so I want to briefly just touch on ketone bodies because when you're doing a ton of fatty acid oxidation, you're making a lot of acetyl-CoA. And what happens if you have too much acetyl-CoA? What do you do? So that's where ketone bodies come in. Yeah, this is a good one. Take a look. So anyway. Okay. So briefly, ketone body synthesis. Okay, this is really important in diabetes. Okay, and we'll talk about that. So where is ketone body synthesis occurring? Good. Yep. The matrix. I heard both. So yeah, the mitochondrial matrix of the liver of the hepatocytes. Very good. Okay. Which molecule in excess starts to stimulate ketone body production? I already gave this away. Acetyl-CoA. Very good. So what is the major function of these ketone bodies? What do they do? Good. Yeah, I heard energy. That's good. So what they are is a soluble energy source. Okay. They can be transported to the brain, heart, and kidneys. Those are the 3 main organs that will use them when you're starving. Okay. So why do we need to convert acetyl-CoA to ketone bodies? Why do we need to do that? Why can't we just send acetyl-CoA wherever we need to send it? It's not soluble, right? You can't transport it in the blood. So ketone bodies are though. Okay. So ketone bodies are a way, they're that soluble energy source that gets sent wherever it needs to go. So I kind of gave this away already. So what's another situation where we produce ketone bodies other than starving? Diabetes. Good. Yeah, diabetic ketoacidosis. Okay. So people with diabetes, what happens is they they can't get glucose into their cells, right? Glucose is stuck in the blood because they don't have insulin, or the insulin isn't working. So they get glucose in the blood, so they can't get energy into their cells. So what they do instead is they start breaking off a lot of fatty acids, and then they'll break those down in oxidation, okay? And then once they've saturated the TCA cycle in the liver, the excess acetyl-CoA doesn't know what to do. And so then the liver sends, okay, let's make some ketone bodies, and that'll be a little alternative source of energy, okay? That's kind of what happens there. Okay, I think we talked about that. Yeah, so the thing with ketone bodies too is if you look at them, acetoacetate and beta hydroxybutyrate are carboxylic acids, okay? These are really acidic. So these ketone bodies acidify your blood, and you can get metabolic acidosis from this, which can be kind of dangerous. And then the thing to know, they like to ask you specifics about these three compounds. So acetoacetate's like your main one, okay? Beta hydroxybutyrate isn't actually a ketone, so it won't get picked up on like a ketone test, okay? Ketone tests only pick up acetoacetate and acetone in levels, okay? And acetone, you can actually exhale that, and that gives it that fruity odor. So people in DKA, they'll come into the ER and they'll be breathing, and it'll smell like fruit. So yeah. So anyway, useful thing to know about acetone. So, okay, any questions about ketone bodies? This is just a very brief overview. You're gonna get this again and again, but I thought it'd be useful to talk about it. Yeah.
Sorry, did you say that on a test you can also pick up aceto?
Yeah, you can pick up acetoacetate and acetone, but you can't pick up beta hydroxybutyrate because in its natural form, it's an alcohol group, not a ketone.
Yeah, this carboxylic acid doesn't really count. So this is a ketone group here. This is a ketone, and there's no ketone here. It's just a carboxylic acid and an alcohol. So a test won't pick that up.
Again, not super important for you guys to know now, but fun fact.
Eventually.
Yeah, what's up?
I believe it is HMG-CoA synthase here, but I'm not
no,
totally
because
sure
it
about
goes
that.
to it's HMG-CoA lyase.
Is it? Okay.
Because HMG-CoA synthase is, it produces HMG-CoA, which is what we're going to go over in a minute with the cholesterol synthesis, cholesterol synthesis. So once you generate the HMG-CoA, it can either stay in the cytoplasm and undergo cholesterol metabolism, or it can go into the mitochondrial matrix via HMG-CoA lyase and become ketone bodies.
Yeah, this would be more the committed step, I think, where once you go through this enzyme, you've made your ketones and you can't go back. So good question.
Any other questions? I know we're kind of going fast, but you guys are just kind of
You guys
killing
are too
it.
smart, so
Yeah.
you didn't spend too much time. Okay, so now we're going to go fatty acid synthesis. Okay, so reverse of fatty acid oxidation. Okay. So it's a really bad joke. Sorry. Anyway, okay, so okay, yeah, so let's see. So where this picture is going to probably give you all the answers, but where in the cell is fatty acid synthesis occurring? Cytoplasm, good. Remember, oxidation was in the matrix. Synthesis is in the cytoplasm, okay, in this case. So what serves as the building block for fatty acid synthesis? Acetyl-CoA, good. Okay, so we have a problem though, because most acetyl-CoA is in the matrix from pyruvate dehydrogenase. So how do we get acetyl-CoA out back into the cytoplasm? Good, citrate shuttle, okay. We convert oxaloacetate to citrate, so we add 2 carbons here, and then that 2 carbon unit gets shuttled out as citrate, and then citrate gets converted back. You then release coenzyme A, or acetyl-CoA actually. You release the 2 carbons and then coenzyme A binds, and you get the acetyl-CoA back. Okay. Any questions so far? This shuttle is important.
I agree.
Yeah. So citrate pyruvate shuttle. I know we're like saying everything's important, but so remember it was the carnitine shuttle for fatty acid oxidation. Here it's the citrate shuttle. Okay. Okay. This is from first aid as well. I like first aid because it simplifies things for me sometimes. So this is just a simplified version of that citrate pyruvate shuttle. Okay, that gets our 2 carbon units out for synthesis of fatty acids. Okay. All right, so which enzyme is the major site of regulation of fatty acid synthesis? It says it up there. Acetyl-CoA carboxylase, good. ACC, okay. So there's 2 up here already. So, but which molecules activate ACC? So we have citrate, And then there's another one. So if we have, think about like a high energy state, what hormone is circulating? Insulin, very good. So insulin actually stimulates acetyl-CoA carboxylase. So if you have a lot of insulin, you have a high blood glucose, your body's like, okay, we have excess energy, we can use that to make some fatty acids. Okay. Now, what inhibits ACC? Okay, so palmitoyl-CoA. And the reason for that is because if you guys remember this 16 carbon unit, this is the product of fatty acid synthesis. So it's like a feedback inhibition. Okay, and then what else inhibits it? Glucagon. Very good. Okay, so low energy state, we don't need to be synthesizing fatty acids. We got to conserve our energy. Okay. So what substrates and cofactors are required? So there's 3 substrates. What do we need? One of them's up there already. Okay, I heard acetyl-CoA and I think I heard ATP. And then we also need CO2. Very good. CO2 because we're adding a carbon. Okay, malonyl-CoA is a 3-carbon unit and acetyl-CoA is only 2. Okay, what cofactor? This is sort of a hard one. Biotin. Good. Vitamin B7. Okay, that's another word for that. So.
Good.
Okay, so same thing with the fatty acid oxidation. There's this 4-step process, okay? And it's important you know the differences. So in the case, we have a question here, hold on.
Mm-hmm. She said,
Do with these citrates. I actually don't know the answer to that question, but I can. Yeah, we can look up that and get back to you on that one. That's a tough one. So, but if they're saying something in lecture, it's probably true. So they're experts. But
we can try and find an answer.
We can try to
for
figure
you.
that out.
We'll make sure that we take a picture of your question and then we'll look it up after this and hopefully send out an email to all of you guys.
Yep. Okay. Anyway, so back to this. So 4 steps of fatty acid synthesis when we're adding 2 carbon units. So it's the opposite. It's kind of the opposite of fatty acid oxidation. But so the 4 steps here are condensation, reduction, dehydration, reduction. Okay?
And make sure you know the orders.
Yep. So same thing. Know the 4 steps and know the order. Don't need to know the enzymes names. They didn't even list them here. And also know that fatty acid synthesis requires NADPH. Okay, that's another big thing to know.
Which is a byproduct of which pathway that we talked about. Nice. Good job.
So our final product of fatty acid synthesis is palmitoyl-CoA or palmitate, which is 16 carbons.
Oh, it's because it's fine.
Oh, there we go. Okay.
All right, let's talk about cholesterol. Okay, so I'm gonna point with my cursor, if that goes away, and we're just gonna do the same game. So acetyl-CoA combines with what in the beginning of cholesterol? Anybody know? Acetyl-CoA. Cool. So
If you don't know the answer, just acetyl-CoA is a good guess.
Yeah, acetyl-CoA is a great guess. So then we do HMG-CoA synthase. What does that generate? I wonder what it would be. HMG-CoA. Huh. So this is what I was talking about earlier. HMG-CoA synthase generates HMG-CoA. HMG-CoA can either stay in the cytoplasm and undergo cholesterol metabolism, or it can go to the mitochondrial matrix and undergo ketone body formation. Let's see. So then what is this one? It's a big star, so it should give it away. HMG-CoA reductase. Yeah, and that's a really important one to know. I would, not just for this week, but pretty much the rest of med school. It'll come up in so many different blocks. So just knowing that HMG-CoA reductase is the target for statins. That's a good fact to know, both now and later. So then between, I'll give you a 50-50 shot. Between insulin and glucagon, which one will activate HMG-CoA reductase? Insulin, yeah. because we have high energy state, we have so much glucose, we're generating so much acetyl-CoA, therefore we're going to want to go down all different kinds of metabolic pathways, including cholesterol metabolism.
The synthesis pathways.
Yes. And then the red one, glucagon. And then this little guy right here.
Is it going to do that?
Oh, just kidding. There's one over here first. Sorry.
Hiding.
Does anyone know what this one is? Squalene. Good job. And then one more right here. It's cholesterol. Sorry. So we talked about this. What is the rate limiting step of cholesterol synthesis? HMG-CoA reductase. And then, how do cholesterol levels impact HMG-CoA reductase activity? It reduces it, yeah. So the more cholesterol you have, the less cholesterol you want to make. So it will cleave HMG-CoA reductase. And then what about insulin and glucagon? We already talked about those actually. So insulin will up or down regulate it. Upregulate. And then glucagon will down regulate it. And so, like we said earlier, insulin removes phosphates. So it activates protein phosphatase, which takes a phosphate off of HMG-CoA reductase and makes it more active. And then glucagon will activate AMP kinase, which will phosphorylate HMG-CoA reductase and make it less active. And this is kind of summarized here. So just knowing that HMG-CoA reductase is more active when it's dephosphorylated, not when it's phosphorylated. And then if you know whenever it's dephosphorylated, it's more active, then you know that insulin is what's going to trigger it to keep going or activate it, upregulate it. Does anyone have any questions about that? Okay. And then I would also highly recommend you guys understand this as well as the next slide that we're going to go over. So how low cholesterol and high cholesterol will impact HMG-CoA reductase and cholesterol synthesis. So I'm just going to walk you guys through this really quick. Pretty much the biggest thing that you need to know is that SRE is going to go into the nucleus and promote transcription factors that code for HMG-CoA reductase. So the way that this works is SCAP is usually an intramembrane protein. This is kind of more detail than you guys will need to know, but sometimes if you understand the process, it kind of helps it click a little bit more. So SCAP is usually associated to this anchor protein. I don't remember the name of it, but whenever cholesterol is bound to it, then SCAP will stay cholesterol bound or will stay transmembrane bound with this SREBP. Does that make sense so far? So then whenever cholesterol is low, cholesterol is no longer binding to this SREBP, and that causes it to disassociate from the SCAP protein. And then that will trigger serine protease to cleave the luminal side, and then a metalloprotease cleaving the cytoplasmic side of this SRE. And that's what allows it to go into the nucleus. So you have to cleave the part of the protein in order for it to be able to go into the nucleus. I know that sounds really complicated. Does anyone have any questions about that? So pretty much just knowing that whenever cholesterol is low, it's not bound to this random transmembrane protein, and then that causes SRE to be cleaved off of the protein and go into the nucleus. So that's kind of a short summary. You guys don't have to know SCAP or SREBP, just knowing SRE is good enough. And then high cholesterol. This one's a lot simpler. So whenever you have high cholesterol, high amounts of cholesterol are just going to promote ubiquitination of HMG-CoA reductase by recruiting a bunch of anchor proteins that ubiquitinate it. So pretty much it just gets tagged for degradation. Does that make sense? Does anyone have any questions? Perfect. So we're going to talk about arachidonic acid metabolism in a second. Glucagon.
Yeah, no, glucagon pretty much always leads to, yeah,
glucagon should always phosphorylate.
that's what it says though. Yeah, that's what there's.
Yeah, they're saying
Yeah.
if there's any exceptions to.
Yeah.
I don't think so.
Okay, yeah, no, yeah, there really aren't any exceptions, not for our purposes. Maybe there are out there in the ether, but not that I've seen, so.
All right, let's talk about arachidonic acid. De nada. I think. Okay, let's talk about arachidonic acid. Sorry. Okay, so what is this enzyme? Do we know?
Starts with a P. PH? PHO? Good.
Yes, phospholipase A2. And what does that generate? Arachidonic acid or arachidonate, yes. And then what is this? This is probably the biggest, most important one out of this entire pathway. COX-1 and COX-2. So there's a difference between COX-1 and COX-2. COX-2 is more important for the synthesis of prostacyclins. And do we know what the function of prostacyclins are? Like, do they promote platelet aggregation? Do they inhibit platelet aggregation? Do they vasoconstrict? Do they vasodilate? Perfect. Yes, they vasodilate and they prevent platelet aggregation. So that means that thromboxanes are going to do the opposite, and they're going to constrict blood vessels. So obviously, we just talked about the role of COX. It's important in generating prostaglandins, thromboxanes, prostacyclins. I don't know if you guys covered this, but how do COX-2 selective inhibitors increase chances of cardiovascular disease? Did you guys talk about that at all? No, not really. Okay. Well, we won't go into detail about it, but pretty much there's just an equilibrium between COX-1 and COX-2. They're both active. So there's an even production of prostacyclins that are important for dilation and thromboxanes that are important for vasoconstriction. So whenever you specifically block COX-2, that increases the amount of thromboxane and decreases the amount of prostacyclin. And that leads to the constriction of blood vessels as well as platelet aggregation. So you're more prone to clots because your platelets are aggregating in your blood vessels. Does that make sense? Yeah. They're not necessarily acting against each other. No, they just convert different, COX-2 is important for prostacyclins. So it's prostacyclins and thromboxanes are not the same thing. They're just like two separate sides of the pathway. Yeah.
So yeah, the interesting thing here is they're expressed in different sites. So that's why they have different roles.
Yeah,
so,
they're not opposites. They're not working against each other at all. They're just producing different products that do different things.
Yeah. Yeah. So I stole this from Dr. Murray. He's going to talk to you guys about NSAIDs and stuff, but most NSAIDs like ibuprofen are pretty equal inhibitors, but like we said, like celecoxib, that's the only COX-2 selective inhibitor left on the market because all of them are cause so many bad heart attack, stroke. So most of them have been withdrawn for that reason. But like ibuprofen, is pretty even. So most of them are because the whole idea was, if we can inhibit selectively COX-2, NSAIDs have a lot of nasty side effects because if you inhibit COX-1, you get stomach ulcers, you can get gastric inflammation. So they were thinking, if we can just inhibit COX-2, we can inhibit the pain and inflammation, and we don't have to worry about these nasty side effects. But there
are other nasty side effects.
Yeah, this was too much to overcome. So that's the reason for that.
So just a primer. You guys will be ahead of the times.
That's.
Okay. Do you want to talk about
Is this me? Sure. Yeah. Okay. Urea cycle. Okay. Okay. So urea cycle. So when we think about the urea cycle, I don't know. Yes. Good. Okay. So with the urea cycle, what we have carbon dioxide and what starting off the urea cycle? Ammonia. Good. And ammonia is the breakdown product of our amino acids, right? So if we're eating a lot of a high protein diet, we're going to make a lot of ammonia, and ammonia is toxic. So we got to get rid of it. So that's the whole purpose of the urea cycle, right? Is to get our ammonia out. So what is this enzyme right here? Very good. Yeah, carbamoyl phosphate. Yeah, CPS. Yeah, carbamoyl phosphate synthetase 1. Yeah, I don't know why it's not full there. And then what do we get as a product? Citrulline. Okay. Yeah, non-intuitive. So so citrulline is our product. And maybe you don't have to know that. I don't know, but I would know CPS1 is important.
Yeah,
Yeah.
so you guys are going to learn something tomorrow. You're going to learn amino acid biosynthesis, and you're going to learn purine synthesis and degradation. And there's a carbamoyl phosphate too that you guys are going to learn tomorrow. So just knowing whether knowing that one is associated with the urea cycle and 2 is associated with purines. That's a really high yield fact to know. I highly recommend you guys know the difference between the 2.
Yeah. And then we have this here, this N-acetyl glutamate. And
Yeah.
oh yeah, sorry. I think you might be right about that.
I agree with that statement.
Yeah, I think this is an oversimplified version here. So yeah, I think I think you're right about that. This
Yeah,
is
you're definitely
oversimplified.
right. I think
So,
it was just trying to skip.
yeah, I think it
down.
skipped a step. It skipped a step. Yeah, that's my bad.
Thank you for pointing that
Yeah,
out.
thank you. Yeah, so we skipped a step here. So, okay, back to CPS1. So we have this N-acetyl glutamate here, and that stimulates CPS1. And so when we think about why that happens, so N-acetyl glutamate, so we have glutamate, right? Glutamate is an amino acid. So high levels of glutamate mean you have high levels of amino acids. You're going to make a lot of ammonia, right? So as we break those down, so high levels of amino acids, N-acetyl glutamate is just sort of a stand-in. You want to run the urea cycle more because you're gonna you need to excrete your ammonia. Okay, so that's the major activator of CPS1 is N-acetyl glutamate. Okay, yeah. So so you guys probably saw, but it's the liver that does the urea cycle. Okay, liver is the main site of ammonia detoxification. Okay, so this is, you can kind of see this from the picture. Yeah. So mitochondrial matrix is where we start. Okay, that's where CPS1 is, and then we go back into the cytosol for the rest of it. Okay, rate limiting step. Yeah, we talked about it, and we talked about the stimulation. Okay, so indicator of amino acid levels. So glutamate and arginine as well. So that's another amino acid. So kind of along the same lines.
Do you want to save that for them to do on their own, or do you want to go through this integration?
I don't know. Up to you. What do you think? We can go
Do you
through.
guys want to go through integration and metabolism together, or would you guys prefer to do it on your own? It's pretty much just like going through which like pathways are in the cytoplasm versus which ones are in the matrix. And yeah, so like we're going to do in the cytoplasm, and then we would list them all off, but they're already listed here. So do you guys want to, I know it's been an hour, so I don't want you guys to like keep going if you guys are tired. Keep going. Okay.
All right, cool. All right, so list me some pathways that occur in the cytoplasm.
Glycolysis. Nice.
Good. What else? Gluconeogenesis. Good. Yeah. Glycogen synthesis. Good. Yep. Fatty acid synthesis. Good. There's one other one.
It's the random one that randomly produces NADPH. Pentose phosphate pathway. Good job.
That's all of them. Okay. What about the inner mitochondrial membrane? There's only one, and we didn't really talk about it. Electron transport chain. Very good. Okay. How about the matrix?
Good.
Yep.
Beta oxide. Yep. And then
ketone bodies. But, yeah, that's good. And then there's a couple like that do both. Like we just talked about the urea cycle, and then gluconeogenesis. Remember, those first couple steps are in the matrix, but mostly in the cytosol. Good. Yeah,
right.
This picture, right? This is our friend. So.
Okay, this is the last slide. So you guys are in the home stretch.
Yeah. All right. So what pathways stem from glucose 6-phosphate?
Glycolysis.
Good. What's the other funny one that we
keep forgetting?
Good.
Pentose phosphate pathway. Yeah.
Yep. There it is. Good. So these are what glucose 6-phosphate is converted into. Okay, in each of those pathways. Okay. What about pyruvate to acetyl CoA? What follows that? Good. And then fatty acid synthesis too. Okay, because we're going back. Okay. How about pyruvate to oxaloacetate? Gluconeogenesis, that's the big one. Yep.
Job.
And then you can also go, remember with our pyruvic carboxylates, we can go straight into the TCA cycle if oxaloacetate is running low. Okay, bypass PDH. How about acetyl-CoA? This is a big one. So TCA, good. Fatty acid synthesis, good.
Ketones, yeah.
What about cholesterol? Last one.
Favorite.
Yeah. AKA flavor. And then citrate. The big one's the TCA cycle, right? I heard that. And then what's the other one? What's the shuttle? Very good. Fatty acid synthesis. Very good.
Job, guys.
Just crushed it. Okay, and that's it for us. So
Yeah. Do you guys have any questions? Okay, so we're going to be sending these slides out, but we also included some slides for your lectures tomorrow, but we didn't want to like overwhelm you with them. And we wanted to forewarn you that the purine metabolism might seem a little overwhelming at first, but it's okay. It's really not that bad. Just take it step by step and remember just the major steps of it. So just make sure you guys don't get too overwhelmed because I know it's Thursday before the quiz. So getting a kind of a heftier lecture can kind of be hard, but it's really not bad. You just have to take it with a grain of salt. But yeah, I think that's pretty much it.
Yeah, feel free to reach out to us with any questions that come up. We'll look at the chat here and we'll try to answer those questions for you guys. Send it out in the email. We'll send you these slides too.
And you guys
tonight.
can do it. It's really not that bad. I know it's a lot of memorization. Try to write out the pathways, quiz each other on which ones are the rate limiting steps and how each substrate interplays with it. But yeah, you guys all knew a lot of stuff. So you guys are on the right track. It's only Wednesday.
So cool. All right. Oh, and there was a sign up sheet going around. I don't know where that ended up, but if you guys haven't signed in, find that.
Yeah, if you haven't signed in, please sign in.
Yeah.
Thank you.
Trying to get a head count. So, and we'll stop.
Yeah.
Let me stop the recording.
It runs all the time because it generates